So many mics. I'll use this one. All right. Good morning, everyone. Everyone's still talking a little bit, so I'm going to try this one more time. Good morning, everyone. Good morning. Wonderful to see all of you guys here this morning, students, instructors, TAs, friends and family. Welcome to the final presentations for the Nanoscale Microscopy course for 2017. My name is Dr. Rita Blake. I am the manager of education here at the California Nanosystems Institute at UCLA. For those of you who don't know me, just a brief intro. So I actually went to school here. I got my PhD here in 2015 in the material science department, did lots of research and outreach in this building, and now I'm here managing all of the different education initiatives that CNSI runs, of which we have many, one of which is this wonderful summer program that you all decided to attend or bring your children to. So welcome, and today will be really sort of the accumulation of all the really hard work that they've been doing over the last 5 days, and you'll, I think, really be impressed at what they've been able to achieve in a very, very short period of time. So just for those of you who have not been here, I'll just give you a very brief introduction as to where you are. So this is the California Nanosystems Institute at UCLA. We're a pretty unique place on campus because we're not a department like most of the buildings here. They're part of a department. We are actually not a department. We don't grant degrees here. What we do is we do interdisciplinary research, all centered around nanoscience. So we're very centrally located within the sciences on campus. We're surrounded by engineering, physical sciences, life sciences, School of Medicine, all of these different programs that have really excellent researchers. Any of them who do anything that's connected to nanoscience will come here, be a part of our membership of faculty. We have many, many faculty here, over 150. And they all come together to do work that is centered around nanoscience, which is the science of things that are on the scale of billions of a meter. So we're talking things that are very, very small, smaller than cells, things on the scale of proteins, DNA, viruses, that kind of level. So it's a really interesting space because we have all of this faculty, again, that come together. We have a huge amount of research space, many of which is dedicated to shared facilities that people from all over campus and outside of campus actually use, that are directed by some of the faculty for this course that actually was working with all of the students for this whole week. We have a lot of inventions also that come out of here because we also house a tech incubator, the only tech incubator on campus. So if you are a researcher here, say a grad student, and you have an idea, you think it's marketable, it has product potential, you can develop it here and then use this space as a place to develop your idea so that it's more ready to then bring out into the world. And so there's a lot of really unique stuff that happens in this building, many of which is, or at least a good chunk of which is facilitated by the faculty of this course that I will now introduce. So the first person I'm introducing, I'm actually going to have him come up here. I told him yesterday he didn't need to come up, but I decided later that that was a lie. And I do want him to come up because he, I think, has really been one of the key people in getting this course started. So I just wanted to let him have a minute or two just to say a few words about the course and why it exists. So the first instructor for this course I'm happy to introduce is Dr. Adam Steed.
Okay, thanks, Rita. Yeah, so I found out about this about 2 minutes ago, so I'll just wing it, which is fine. I'm usually relatively comfortable winging it. So welcome to CNSI. Rita gave a nice introduction of what it is that we're doing here. It is a research institution mainly focused in the day-to-day. But as not being a department that grants degrees, we do engage in a lot of different educational activities. We host all sorts of meetings and symposia for faculty. We have training programs for graduate students. And we have started to develop over the last few years some undergraduate programs, specifically that this course is one of the few that is based upon. So we do actually teach a class. The 3 instructors for this course teach a course in the fall called Biological Microscopy in the Department of Molecular Immunology, Microbiology, and Genetics, I believe, if I remember the whole acronym correctly. And it's really trying to convey to the next generation of scientists and researchers the really empowering capabilities that technology provides to discovery. And that's something that is traditionally not a normal course. You wouldn't find this course delivered in a traditional academic department. The interdisciplinary nature of the researchers and scientists at CNSI provide a unique opportunity not only to do the research, but to develop coursework that can really enable our students here at UCLA to have a different perspective on what is going to allow them to be the next innovators. And what we've been also in parallel developing over the years is a perspective that it's not just about an opportunity to teach university students, undergraduates, graduate students, and to collaborate with our colleagues, but really a responsibility to engage with the community more broadly through all of the outreach efforts that Rita's been associated with and manages throughout the years, and in developing coursework through the UCLA Summer Sessions program to bring the even further next generation, high school students and middle school students, to UCLA to really expose them to what the leading edge technologies are and where the field is going. So this course is one of 4 that we deliver, but sort of back to back to back to back. Rita's summer is probably the most complicated period of the year. Most people think that it slows down in the summertime, not around here. And this is going to be the first one. I believe this is our 3rd year we've done this program, and we're really, really excited about the group this year. We've sort of dropped them into the deep end and immersed them in lots of different technologies and concepts related to both what it is to do research and to understand the world at nanoscale dimensions, and how we use advanced microscopy and technology to really explore not just basic research, but to really address the grand challenges of the 21st century that face us all. So we're looking forward very much to hearing what they all came up with. I know it was a really busy, hectic week. I hope that everyone enjoyed themselves, and it's been a pleasure to meet and work with all of you. So with that, I'll turn it back over to Rita and just thank you all for your energy, and for the parents who are either here or listening for the opportunity to send these bright young minds to us, because it's inspiring to see the next group of people like myself. I flash back and realize that it's been almost 26 years since I was their age. So it's good to see a new generation really coming in so passionate, and we're looking forward to seeing what they have put together for us today. So thank you all, and welcome.
He is good at winging it, isn't he? So I also want to introduce, and he doesn't need to come up, but I'm just going to introduce him, Dr. Laurent Bentalila, who's also one of our instructors and also a researcher at this institute. So let's give him a hand as well. Our other instructor is Dr. Hong Zhou, and I am not sure that he is here at the moment, but he will probably be dropping by in a little bit. He is also, again, a researcher here, a director of the Electron Microscopy Lab. I will say that Adam is the director of the Nanopico Lab, which is focused on scanning probe microscopy, and Dr. Bentalila is the director of the Advanced Light Microscopy Lab. We also had a very, very invaluable resource this year in the form of Michael Lake, who is a graduate student here at UCLA and was the teaching assistant for this course. So we'll give him a hand too. He's in the Department of Chemistry and Biochemistry, and we've been very lucky to have him because he's very knowledgeable and quite skilled at all of the techniques that the students were learning about today, or in the last 5 days, excuse me, as well as 3D printing, which was a new kind of fun element that we included in the course this year. So it's really been great to have him and to work with him. We've also had a number of people working behind the scenes, helping to ensure that everything went smoothly throughout this week. And I don't have the time to introduce and thank all of them, but amongst all of the CNSI staff that really does deserve a big thank you, I will specifically highlight Dr. Elaine Morita, who is our program coordinator, and really has been instrumental in keeping my brain functioning well, considering all of the different summer programs that Adam was mentioning that I'm running. So Elaine, thank you so much for everything also. So just to give parents, again, a little bit more understanding and to let you guys kind of reflect on what you've been doing these past 5 days, I kind of wanted to show you guys a little bit about what they've been doing. So there was really 2 goals with this course. One is to expose you guys to advanced microscopy. So really looking at not just, a little light microscope you might see on a bench in your classroom, which is pretty common, but to actually see like how small can we see, how deeply can we probe into these materials, these new questions that are coming up into the 21st century, and how can we use these techniques to really address these problems, these grand challenges that Dr. Steed mentioned. So with each technique during each day, they had a number of things to think about. One, they had to do their homework every night, which was to write about the technique that they were working with and relating it to one of two grand challenges that we presented them with. These grand challenges are challenges that have been identified by senior scientists in the nation, as well as the president at the time that these recent ones were created, President Barack Obama. So they're very, very, very big, very difficult challenges to grapple with. And we really asked the students, as you guys probably know, since we sent your students, your children homework before they got here, to read about these grand challenges, and then used microscopy as a framework to think about how these challenges are going to be addressed. So they had to think about these things every night and write about an idea or something that inspired them about the technique that they learned that day in relation to these challenges. And then also they were working on their final projects that you guys are going to see today, where they're really thinking about how the instruments that they've been working with address these grand challenges, or what new instruments need to be developed in order to better address these grand challenges. And they're going to be working in groups of 3. And just so that the students have a reminder on this, you guys are going to have 7 minutes to give your presentation. I will be timing you, so please be on time. And then we'll have about 3 minutes of Q&A, and Q&A is open to everyone. So obviously we're going to ask you guys questions, but anyone in the audience, parents, your peers, anyone is free to ask questions. We want this to be a truly sort of open, kind of informal Q&A. So it's not meant to try to be mean or anything to you. We just want to see what you've learned and kind of get your understanding of the topic. And this was their schedule every day. You can see just by looking at it how packed it was and how much we really sort of got into with these students. So we really did a lot. So the first day, they were really given sort of a broad introduction. So we had an introduction into what is nano, because we're talking about nanoscale microscopy. They got to learn a little bit more about the CNSI, and they got to take a kind of sneak peek into all of the different research facilities in here in the incubator, which most people don't really have access to. They got 2 very interesting introductions to the 2 grand challenges that we addressed. So they got to hear from Dr. Jeff Miller, who is actually the director of this institute, talking about the microbiome. And we had Dr. Wendy Walwyn talk about the Brain Initiative, and they both kind of referenced these initiatives and these challenges in the context of their own work. And then we got into some DIY microscopy, where they actually built their own microscopes using webcams and flipping the lens. The next day, they did work into what is called fluorescence microscopy and also confocal microscopy, where you're using fluorescent dyes or fluorescent proteins to be able to tag things in the body and to be able to, or in cells, or in animal models, and to be able to get these beautiful, really wonderful images, and to really be able to see with pretty good specificity things that are going on at the cellular level. And we also gave them a very challenging journal article, which they all, I think, tackled quite well considering the difficulty. And we started to work on some 3D printing. And then Wednesday, they got into electron microscopy, so starting to see at even smaller scales what can be imaged. And they worked with Dr. Hong Zhou and Dr. Peng Gi, both in the Electron Microscopy Lab. And then just yesterday, they were working with Dr. Steed, looking at scanning probe microscopy, so using touch instead of using light or using electrons. So a whole different way of seeing things, a whole different kind of paradigm for visualization. And then we've got today. So they've really gotten kind of this whirlwind introduction, and just by the sneak preview that I got of working around with them yesterday and seeing their slides this morning, I'm really excited to see what they've come up with, because it was no small task what we asked them to do. And I think they all met the challenge quite well. So with that, I am going to stop talking myself and start inviting all of you guys to come up and give your presentations. So thank you very much, and I look forward to seeing what everyone is doing. All right, so group one. Oh, sure. Oh, is it fine? All right, you could do it. I have faith. All right.
Can we start? Yes, go ahead. Okay, so I'm Jonah Metalcats, this is Ian Livingston, and this is Sartaj Ball, and our presentation is about the connection between the gut microbiome and the immune system. So a little background on this is that a microbiome is a community of microbes, which are microorganisms found in the body and nature, and we are focusing on the human gut microbiome. And this kind of focuses on the grand challenges we've been learning about this week, which is to study the microbiome in humans and figure out how the microbes there interact and how we can use this to impact human health.
All right, so our question is how do gut microbes impact the immune system? Scientists have found a link between microbes in our gut to the immune system. The importance of this is to improve vaccine efficiency and discover the cause of ineffective immune systems in our population. Our proposal is to further studies on the microbes in the human gut in order to see the full effect they have on the immune system. Our plan, Our plan is to approach this issue by comparing microbes of the healthy immune system to microbes in the unhealthy immune system by using confocal microscopy.
So for our method to approach this topic, we thought confocal microscopy would be the best. So just a general description, confocal microscopy, it's shown in this image here. It has a pinhole right next to the lens, which blocks out the light above and below the sample. So it allows you to make three-dimensional cross-sections, so giving you a more physical rendering, so it lets you look inside other samples that you have. It's particularly useful for 3D rendering and observation of samples below their surface, which is often what you need to find out exactly what is causing the deficiencies or strengths in immune systems. And links have been found between beneficial microbes and the production of T cells. And hopefully soon they'll also find links between B cells as well. T cells are cells in the immune system that directly kill disease cells, while B cells produce antibodies which counteract diseases that have been introduced into the body already.
Okay, and then moving on to social context, which is basically how this can be applied to the general population outside of the lab. So we already know that gut microbes affect vaccine efficiency, and further understanding these microbes can help us improve these vaccines to a higher level. And then targeting the immune system through these microbes can enhance research and prevention for diseases like HIV and AIDS, which weaken the immune system. And we know that gut microbes are influenced largely by your diet and what you eat. So hopefully studying the microbes in the gut can help create diets for the public that can strengthen their immune systems.
So there's been multiple studies in recent years exploring the connection between gut microbes and the immune system. So in 2014, the University of British Columbia began researching asthma, allergies, IBS, and diabetes. So they found initial signs that the composition of microbes can lead to failure to fight these diseases. The Weiss Institute of Harvard built upon this and focused specifically on IBS. So they have presumed links that microbes, epithelial cells, and the immune system are related, but as of now, they have no definite conclusions on why. So that's a field that can be researched further in the future.
So our project proposal is to continue using confocal microscopy to study the human gut microbiome. Using our data, we want to continue making links between the gut microbiome and the immune system and build off of preliminary studies on non-human subjects to reach a cure. Our main goal is to fully comprehend this connection in humans and use it to our benefit.
So as of now, there are multiple challenges in approaching this problem. So while it's more straightforward to look at similar microbes in healthy or particularly weak immune systems, it's very difficult to figure out exactly what gene causes the strength or the weakness. So once the particular microbes have been isolated, you have to find what genes are present using confocal microscopy, which presents a challenge. An Imperial College paper outlines the link between microbes, diabetes, and obesity within mice, but they have yet to bring this particular instance into the human scale. So most early testing begins with animal test subjects, but a challenge is that it's difficult to convert from the animal test subjects to humans as well. Yeah.
Okay, so to wrap this up with the discussion, our current proposal focuses only on the associations between the gut microbes and the immune system and finding remedies for the negative effects caused by unhealthy microbes.
Our future direction consists of increasing our scope of research about gut microbes to include all the major body systems of the human body, so cures can be found for any microbe-related diseases. These goals can be achieved by using confocal microscopes to image the full three-dimensional structure of the microbes.
And then, so we have our contributions, which slides we talked on and worked on, and then the references for our presentation. And we can open it up for questions now if anyone has them. Well, for the example about the mice and obesity is just, I think, to show that a lot of times there's links found between the microbes in animals and their immune systems that we can try and connect in humans. But for human testing, it's mostly done. So if someone's getting surgery on their stomach, you can take a sample from there and use it. But also it's pretty easy to test mice, and because that they found that link, we can hope to find links like that in the future on human test subjects.
So when you guys look at the microbiome, do you expect to find patches of individual microbes, or what would you expect to see as the structure of the microbial community inside of something like the gut?
Yeah, so usually the microbes are specific to each of the different regions of the body, and so we're focusing mostly on the stomach and the digestive tract, so the gut. And we, in general, because it's so specific to the area, that in the digestive tract it will be more spread throughout the body. You can test for what areas they are in mice particularly, just because of the question before, the prevalence. But we don't assume to find one exact small grouping of bacteria here and over here. But research is still being done to figure out exactly what amounts of bacteria and what types of bacteria are throughout people, and also depends on the person. Everyone has different microbes, so that changes a lot of different things about you. So it's really variable.
I have a question. So when you were doing research on the links between the microbiome and the immune system, did you find anything about different microbes affecting different systems? And do microbes only affect kind of one system at a time, or are there microbes that might, a single microbe that might affect multiple different systems in the body?
Okay. Well, we found that in humans, like especially like we focused on the gut microbe because it affects so much of your other parts of your body. Is that what your question is?
So I guess my question is like, so there might be links between like X bacteria and the immune system. Would that bacteria also affect other systems in the body? Like if it affects your immune system negatively, might it also affect something else, maybe even positively?
I mean, yeah, that's possible because like especially in the like gut digestive tract that we were focusing on, it affects so many other things. Like it can affect like digestive, like mood, all these other things other than your immune system. So yeah, that's possible.
Other questions?
I'll ask a technical question. So if you image the three-dimensional structure of a microbe, right, like what do you hope to associate with the actual immune response or deficiency of a person from that knowledge gained, right? So you're going to image the microbe, you see what the structure is. What's the association or the linkage that you want to look more deeply into with respect to the actual immune function of the patient from where those microbes came from?
Well, you could like look at the structure of microbes, like we said in the presentation. You can compare the structure of those microbes to microbes with a person who has a healthy immune system. So within that, you could look at the differences between them and figure out like, oh, so this part of the structure is odd, it's shaped a little different.
Are there indications from the literature that structural differences in the microbes have been associated with these changes, like the examples that you gave of previous work? Were they looking at the structural elements of the microbes themselves, or were they looking at other aspects?
Yeah, so there's a study done looking at diets especially. So it was comparing the zinc intake of mice, which was linked to CD8 cells within the immune system, it's a lot like B cells, it produces antibodies. So often it's not the exact, physical layout of the bacteria, it's which are most prevalent in comparable test subjects, and also the diet, which is an extremely large part of what the digestive tract entails.
Students? You guys can't ask each other questions. It's a good thing.
Yes.
After those, like in your discussion about future areas to investigate, what are some of the other areas of the body that we would like to go to after you've done imaging microbiome stuff like that?
Well, we just like to do all the major systems of the body, so like the respiratory system, the circulatory system. Like microbes are pretty much everywhere in your body, so and it's not just a correlation between gut microbes and the immune system. It could be other microbes affecting other immune systems. So hopefully with this research we can build upon it to other systems as well.
Let's maybe take one more question. Yes.
Why did you guys choose the gut specifically?
We chose the gut because it's so, like, there's already been discoveries that link it pretty heavily to the immune system, and we thought that it was super interesting that, like a microbe, this tiny thing in one part of your body could completely affect something else in your body. So that's really why we chose this for our project.
All right. It can definitely be scary to be the first ones up, so thank you guys. Nicely done. All right, group 2.
spacebar? Let's go. Yes, maybe spacebar can work too. Okay, we'll start. So, hello everyone. I'm Jason Lee.
I'm Oscar.
And I'm Alex Polvada, and we did our project on how can we use microscopy to better understand the plaque buildup in brains of Alzheimer's patients. So we're going to use fluorescence microscopy to locate where the plaque builds up in the brain, and then we're going to use electron microscopy to examine the structure of the proteins of beta amyloid. The relevance of this is Alzheimer's is one of the most predominant senile diseases, and by analyzing how the 3D structure of the protein looks, we could possibly develop a cure. From the data, we hope to understand why the plaques build up and where they build up, and then create a cure that can eliminate Alzheimer's.
So some of the social ramifications of research into Alzheimer's is that those affected by the disease tend to lose their memories and then eventually even their identities. And is it mic? Okay. And this especially affects the families of those who are affected by Alzheimer's because the affected basically no longer recognize their family, and also constant care is required toward the later stages. And also there was a 71% increase in deaths to Alzheimer's in the US from the year 2000 to 2013. And the number of seniors in the US projected to have Alzheimer's by the year 2025 is going to increase from 14% to as much as 60% in certain states.
So as an overview of the equipment that we're going to use for our research, we have fluorescence optic microscopy on the left. It functions by exciting an electron, which emits a photon, a light particle, and back into the eyepiece. That's how you see the image. And we have the electron microscope on the right, which features electron beam shooting through the sample onto the fluorescent screen. And what you see, the image that you see, is basically a electron density image of the through the sample. So now Jason's gonna elaborate into how we're gonna specifically use those equipment to foster our research.
So first of all, let me talk about the particular form of fluorescent microscopy. We propose to use a method called Sted Microscopy, which basically the premise of fluorescence microscopy is that a laser is shown onto the sample which has been prepared with fluorescent markers. This laser contains photons which excite those markers, and those markers in turn produce their own photons, which are then captured by a detector. With Sted microscopy, a secondary laser in the shape of a ring, as you can see in that middle picture on the bottom row, is sort of wrapped around the main excitation laser. This causes that main laser to be reduced to a much narrower diameter, which can dramatically increase the resolution of the image, enable us to image in far greater detail and at far smaller sizes. So how this optical fluorescent microscopy will be applied is that we will use primarily antibody quantum dot affinity pairs in order to mark our specimens, as our specimens are going to be primarily protein-based neurotic plaques in diseased brain tissue. The antibodies bind to specific proteins, and the quantum dots, which are made of nanocrystal heavy metals, shine very brightly when excited by the laser, which can enable us to have a much brighter contrast in our image. This fluorescence microscopy can help us compare known healthy brain tissue with our samples, which can confirm the presence of beta amyloid plaques. The electron microscopy phase can enable us to directly image and analyze the structures of those proteins, of misfolded proteins inside the neurotic plaques, which can enable us to learn a lot more about how these, what exactly went wrong in the formation of these proteins and what can be done to stop it. So some of our primary challenges that we will face is finding samples that will correctly match the conditions inside the brains of the humans with Alzheimer's. One of the ways we can get around this is by using brain tissue obtained via autopsy, but the main drawback to that is that the brain tissue could be potentially degraded by storage and chemicals.
So for the literary review, we're gonna run over the foundation's published papers that serve as the foundation of our research. So as you can see on the chart, since we're primarily interested in plaques, so it's colored in green. We have different mice species in columns. And as you can figure out, they fall pretty uniformly and consistently between the time period of 9 months to 12 months. That's how we figured out our test subject and our the specific time frame that we want to work with.
Some other research that we did was we used articles to find out that the beta amyloid protein was the one that was causing the plaque buildup in the brain, and which is the one that blocks the synapses from firing and causes Alzheimer's. Another thing that we found was most of these articles did not deal with the actual structure of the protein. Rather, they analyzed the effects, so that's why we're interested in getting a 3D image so we could learn about the proteins and see how they fold so we could develop a cure. Okay, future direction. We hope to use the electron microscopy to make a 3D model of the beta amyloid, understand it, see how it works, and then hopefully develop a cure and test different medicines to see which ones work best to ultimately end Alzheimer's.
So here is a list of the articles that we have referenced in order to compile our PowerPoint. And
Thank you.
thank you all for listening.
Very interesting, first of all. And what I like is you're presenting two different approaches to the same project, which is really interesting, right? Fluorescence microscopy and electron microscopy. So since you are doing so, I'm guessing that you are expecting to extract different information from the two techniques. So what I would like to ask you is, what kind of information do you expect to get from fluorescence microscopy approach, and what would be the different information you expect to get from electron microscopy?
For the fluorescence, we're wondering mainly where the plaque builds up and seeing how, where and why it builds up, and also just to analyze the difference between the healthy beta amyloid and the unhealthy beta amyloid. So then we could extract and identify the unhealthy ones, which we could then put in the electron microscope and analyze the unhealthy proteins to see how they form and really see how they react to different medicines and see if we could reverse Alzheimer's.
So on your future direction slide, or your conclusion slide, you said that you would identify the structure and then we could start to look for a cure. So after you do have the structure, then what would be the next step that researchers would take?
Well, we could try to analyze the protein, see how they bind, and see, maybe get an idea of what medicines we could use to start reversing it.
So you have seen the structure between a healthy and a defective one, right?
Yes.
What's the, can you kind of tell us the difference or?
Well, unhealthy amyloid beta proteins tend to cluster in a form of a plaque in the brain. So that's the primary reason why we want to study the locations. Once again, the location, their locations, and then further on move on to study their interior structures to develop a cure, as aforementioned by my other fellows.
So unhealthy one tends to like cluster together and stuff.
Yes. Yes, and block the neuron transmission process.
Other questions? I saw another hand raised over here. Yes.
So you said you're going to be testing on mice 9 months to 12 months, right? And so why did you choose to test on them? Like what similarities do they have to the human brain that you're hoping to exploit? Like do mice also contract Alzheimer's as well, or why are you looking?
Yeah,
at these
stuff.
subjects?
They contract Alzheimer's, and it's more of like a moral dilemma because it's kind of, if we try to use a actual patient, then that would be unethical. And then if we try to use the autopsy one, then the chemicals can interfere. So right now the mice seems like the best solution, but we are going to try to maybe use the brains of autopsies of deceased patients, so we could get like a 2-scope to see how one is like right away without chemical treating with the mouse, and then one with the human with chemical treating to get a more comprehensive idea of how the proteins work.
One more question?
I got one more about the structure. Now, in the future, are you guys looking into destroying that, or to rebuild it, or something like renovating it? You know, once I try to fix the structure, or just completely destroy it.
One of the more immediate fixes we predict could be done to dissipate these plaques is sort of develop a different compound that can prevent those beta amyloid proteins from sticking together in the first place. And sort of a compound that binds to the beta amyloid prevents them from binding to each other in turn. With greater knowledge of the structures, we can maybe go a step further and identify the cause of why those beta amyloid proteins misfold and misform in the first place, which would prevent any sort of plaques from forming and solve a great problem in this research.
Well done. All right, group 3.
All right, so for our project, we chose to focus on the BRAIN initiative, and the goal of the BRAIN initiative is to map the brain and get a more thorough understanding of how the brain works on the nanoscale. We specifically chose to focus on the neurological disorder Creutzfeldt-Jakob disease.
So Creutzfeldt-Jacob's disease is a rare, rapidly progressing brain degenerative disease. And we know that it's caused by prion protein mutations, but we really don't know a lot about these prion proteins themselves. So, for example, we don't know a lot about their exact structure or their exact purpose. So we hope to design an experiment that would use different methods of microscopy to do a side-by-side comparison of healthy prion proteins and infected prion proteins. So this would help us to gain a better understanding of the prion, which is essentially our goal, because this would lead to better methods of diagnosis in the future. And it would also address the BRAIN initiative because it would help us to better map the brain in more detail. So what exactly are we trying to answer? So our fundamental question is, what are the fundamental differences between the cell membranes and nuclei of healthy prion protein cells and infected prion protein cells? So in this image, you can see that this is a really simplistic model of the prion protein cell. That's because scientists really don't know much about it right now, and that really isn't helpful when you're trying to solve something like Creutzfeldt-Jacob's disease. So answering this question would really help us to gain a more complete understanding of CJD, and it would also help scientists to develop more effective methods and would lead to improved treatment later on.
Okay. Creutzfeldt-Jakob disease is a rare, degenerative, rapidly progressing, and invariably fatal brain disorder discovered in the 1920s by German neuroscientists Hans Gerhard Creutzfeldt and Alphonse Maria Jakob. This disease is caused by misfolded prion proteins building up in the brain, and these mutated prions infect other healthy prion proteins, eventually leading to the abnormal prion proteins destroying their neurons in the brain, thus leading to a wide array of symptoms. In the early stages of CJD, symptoms include failing memory, blurred vision, behavioral changes, and lack of coordination. As these symptoms are similar to other more common brain disorders such as Alzheimer's, some patients with CJD can be wrongly diagnosed. But the later stages of CJD have more extreme symptoms such as dementia, involuntary movements, blindness, and weaknesses of extremities.
All right, so prion proteins, a mutation in them is what causes this disease, but scientists don't actually know the function of prions when they're healthy and functioning normally. So what they do know is that they're highly expressed in neurons and that they play some sort of regulatory role in the nervous system, though they're not exactly sure what it is. In a study done with mice where they had nullified the prion gene so it could not perform any function, just wasn't working properly, they noticed that the mice were more prone to oxidative stress and unnecessary apoptosis, which shows that, like, once again, they are necessary because they lead to a decreased health if they're not there, but they don't know why that is. They also performed a study with zebrafish embryos where they used green fluorescent protein to tag the prions, and they noticed that the prions tended to accumulate in places where the cells were in contact with each other, so that could mean that they may play a role in cell-to-cell communication. And also, in another study done with zebrafish, they nullified one type of prion protein, and in this stage, the zebrafish embryos did not make it through the gastrulation period. And when they tried it with a different prion protein, they made it to early larval stages, but most of these zebrafish had serious morphological deformities. Oh, and these pictures are both trying to show how the protein changes from normal to when it's mutated. But if you notice, they're not exactly the same picture because scientists don't know for sure the structure and how it works.
So the potential impact. So there are some pretty significant potential impacts. One is, of course, a better way of diagnosing CJD as well as other prion diseases. So it could also accelerate advancements in the research of other prion diseases like Alzheimer's, for example, and it would bring us one step closer to a cure of all of these diseases.
Currently, to diagnose CJD patients, doctors perform numerous tests such as magnetic resonance imaging, electroencephalography, neurological and physical examinations, along with cerebrospinal fluid analysis, which is extremely painful. And EEG is the measurement of electrical activity in different parts of the brain and the creation of a visual trace of that activity. And the cerebrospinal fluid is produced by choroid plexus in the brain and is absorbed into the bloodstream to deliver nutrients to the brain.
So the types of microscopes we're going to use in our design are the cryo electron microscope. And so this one is a cryo electron transmission microscope, which means that first the sample is going to be frozen before it's put into the microscope to create contrast, and then electron beams are going to pass through the sample, and the microscope is going to read them and then create an image from that. The second kind we're using is atomic force probe microscopy. And in that one, a needle probe is ran along the surface of the sample, so it actually comes into contact with the sample and measures its elasticity by touch to create an image. And these, the one farthest to your right is the electron, and the one closer is the atomic force probe. So for our plan to address this issue, we're going to take spinal taps to attain spinal fluid from patients with CJD and some willing participants who are willing to subject themselves to getting a spinal tap, which, yeah. And so we're going to perform the cryo electron microscopy, and we're going to look for any changes in the structure of DNA. So specifically, we're going to see if the DNA changes locations within the nucleus or if the nucleus has any significant changes in size or shape. And in the atomic force probe microscope, we're going to analyze the surface texture of the prions and see if maybe there's any protrusions or indentations particular to the CJD ones that are not in the healthy ones. And hopefully, if we understand the structure of those, we can understand the structure of the cell membrane of cells infected with it. And these, on the top is an electron microscope, and on the bottom is an atomic force probe microscope.
So there are a few challenges with this approach. The major challenge stems from the fact that CJD is really rare. So this would mean that it would be difficult to find patients who would be willing to suscept themselves to spinal taps, of course. And then also it would be really difficult to find definitive patterns because we would have a really limited amount of data with limited patients. All right.
That's all. These are our references.
Oh, and also CJD kills only less than 1,000 people in the U.S., so it's kind of a small number in comparison to the population of the U.S. Other prion protein diseases include Alzheimer's, like the other group talked about, and Kuru, which is found in Papua New Guinea, that's spread through cannibalism, and it's infectious.
So you mentioned the challenges of getting samples from humans. So what would be the alternative to using human samples?
Yeah, if we couldn't use humans, we would probably use zebrafish because they're easy to look at under a microscope, and they have similar DNA to humans.
Is there a mouse model?
There's been trials done on mice using it, so that's also an alternative you could use. I would say either one of those.
How come you don't use like other primates?
Just because in testing it's easier to use smaller animals and more like cheaper, readily accessible animals. I mean, and it's like, I don't know, you run into more ethical issues when you're using larger animals for testing. So.
There's also another factor, which is that Kruetzfeldt Jakob develops over a very, very long period of time. And this is also one of the challenges. It takes about like 30 years before you develop the symptoms. So if you use an animal model that has a lifespan that's much shorter, you can expect to see the symptoms much faster.
That's why fruit flies are used a lot. Kind of. Yeah. Well, you increase it to like 6 figures, I'll do a spinal tap.
You got it. Done and done.
So, I have a question. So, why did you think about looking at the location of the DNA? What would be the effect of different locations?
Well, because we know so little about it, DNA is just like the best starting point we could think of, because if we notice like a difference in structure, it could maybe be due to figuring out what kind of mutation occurred in the DNA, or like what kind of secondary structure changed in the protein, and that would be a good basis to start further research on and possibly develop a more accurate way to diagnose people so they don't have to go through such extensive tests.
Yeah, I can't really.
Yeah, can you say that louder?
Are prions completely resistant to chaperone and repair?
The studies.
Yeah, well, they are because, like you said, they accumulate for years and years. It's called an incubation period, and it just, I mean, they can't be stopped, and they don't contribute symptoms. So, for some reason, yeah, they go unchecked.
So like with diseases, since you mentioned food, so why would you look at genetics if it's like an infectious disease that's acquired?
Only Kuru is the infectious disease. This one's like sporadic. Mostly 85% is sporadic CJD, and the rest is like familial.
Yeah, you can get it, but it's usually the only way to get it between people is by like having a tissue, like if you were getting an organ from someone or some sort of tissue-to-tissue contact.
Yeah, or if you were eating like bad meat, you get it too.
So in the case of Kuru, since you mentioned it, it's not really contagious. I mean, the only way the pygmy of New Guinea, which has been reported, got the Kurus because there is a ritual out there that is to eat the brain of a person that passed away, right? And it turned out that there's a high prevalence of Creutzfeldt Jacobs in that population, and this is how the disease has been passed on, actually. So it's not really infectious in the common sense. It's a very unique.
Isn't that kind of like the way mad cow disease is done?
Yeah, it's just,
Yeah.
it's the same thing.
No, it's the same thing.
Essentially.
Oh, okay.
There's one more question? One more question.
I have one quick question. You mentioned earlier that it was rapidly, that it spread kind of rapidly once you have it, but the incubation is slow, is that?
Yeah, the incubation is slow because as the protein mutates, it can affect other proteins, but it takes a while for that to happen. The symptoms don't usually display themselves till like a year before. Once you're diagnosed, you usually have less than a year to live. it's pretty bad. Like you don't really know until you get symptoms, and then it gets really bad really fast.
I think is it a quick question? It's a quick one. About 2 weeks ago, I was listening to NPR in the morning, and there is an ongoing study right now. A woman had the disease, and they were talking about genetic differences. So it's kind of like an up and coming big topic in science because of the ramifications for other brain diseases. So.
Oh yeah, because like it's a type of prion disease which covers a lot of different things, and it's just like a step forward in understanding like dementia and deterioration of the brain. So research could be like applied or relate to other diseases as well.
All right, thank you guys.
All right, group 4.
Hello everyone, my name is Amanda Jorgensen.
I'm Erica Liu.
And my name is Hope Rvetche, and this is our presentation on the impact of microscopy on neurological and psychiatric disorders. So, how does current fluorescence microscopy affect neurological and psychiatric disorders? Well, many people around the world are impacted by diseases such as Alzheimer's, Parkinson's, depression, etc. And the Brain Initiative has done years and years of research on this. However, we believe that extensive use of fluorescence microscopy can bring us closer to cures for these diseases. We believe that a pre-diagnosis approach would be extremely effective and would enable us to identify any signs of the illness before the symptoms start setting in. So fluorescence microscopy is a type of microscopy that enhances species and creates clearer images for thorough examination. So the process goes that the use of dyes as well as a high energy light excites the specimen, which results in the specimen emitting a low light energy, which then produces an image that is cleared and has intensified features for extensive examination. So this type of microscopy has already been used in multiple sciences, such as the neurobiological sciences, and so we believe that a neuropsychiatric focus will expedite the process of finding cures for these diseases.
Making a breakthrough in how we treat neurological and psychiatric disorders with nanoscale microscopy has the potential to change the lives of many. According to a study made by the UN World Health Organization, around 1 billion people are affected by these disorders, and that's around like 1 in every 6 people in our world's population. So with such a large number of people, we think that being able to use nanoscale microscopy will be able to change the lives of the many. We hope that being able to utilize this technology in pre and early stage diagnosis will be able to treat them from the very beginning, and also discovering elements that are related to the disorders like gene and chromosomes. So there are multiple types of dementia which make it difficult to identify the exact cause of the illness, and if we're able to use this technology to look closer into it, we want to be able to look deeper and find out what can differentiate them.
So right now, not much is known about how these illnesses are caused or how they can be cured. But what we do know is that these psychiatric illnesses, they progressively get worse as time goes on. So right now, treatment focuses more so on slowing down the progression and kind of stopping it at where it's at. But so far, we haven't been able to cure all of the effects of the illnesses that have happened so far. And so right now, scientists are attempting to pinpoint the certain brain circuits that are associated with these illnesses by stimulating regions of the brain to better understand how these illnesses kind of affect the brain as a whole, which may lead to a more effective treatment. And then in 2016, Professor Marcus Sauer from JMU and his team, they wanted to tag proteins that were related to these psychiatric illnesses. And so they originally used antibodies, but the issue with antibodies is that because of their size, they are much larger than the proteins they wanted to tag. The images that came out were not clear. They were blurry images, and so they were not able to get conclusive results from them. And to overcome this, they teamed up with the University of Copenhagen, and they created peptide probes. Peptide probes work because they are much smaller than the antibodies that were originally used, and therefore the images that result from it are much clearer, and they could get more conclusive results from it. So far, the results have been promising.
So essentially, our project takes a pre-diagnosis approach, meaning that we want to pinpoint the disease before the symptoms actually start setting in. And so to do this, we'd be using peptide probes to identify certain brain regions, as well as proteins, cells, genes, anything that could point us in the direction of explaining how and why these diseases affect people as they do. And so peptide probes would be very effective because they're small enough to produce very clear images, as well as attaches very effectively to the specimen in question. And so we believe that this can make significant progress that will positively impact the BRAIN initiative and the research they've done in the past. And so with these innovations comes setbacks. And so one of the possible obstacles that we'd be facing is that current fluorescence microscopes cannot provide spatial resolution below a certain diffraction limit. And so the difficulty comes in that different species have various limits, making it very difficult to pinpoint one exact diffraction limit for all species. Another obstacle is that observation of living specimen is much more difficult than that of the deceased. So normally when observing a living specimen, it would result in a either too faint or too bright non-specific autofluorescence, making it practically unusable and inobservable for research.
And just to summarize, we want to use fluorescence microscopy and peptide probes to pre-diagnose brain disorders, and this will address the BRAIN initiative grand challenges. And we want to aim to help the millions of people that are affected by these neurologic and psychiatric diseases.
Okay, and then contributions and references. So we're opening the discussion to questions. It's very similar to what we're trying to do. They're using it to identify certain proteins and the effects of it on the body. And so we believe that if we were to use the same peptide probes on neurological and psychiatric disorders, it could have the same effect in identifying what and how things cause effects in the body.
Yes. So if you're using it to like pre-diagnose, how would you be getting the samples to like test it on? Would you take it like from a spinal tap or like a biopsy?
Well, what we could do is that we could use patients who already have the disorder and see what they kind of have in common. And then maybe we could use maybe one of their relatives or something, since it's likely connected to genetics. Maybe we could see if they have that same gene or chromosome as well.
What got me interested is that PTSD and all that stuff. I thought it was mostly more environmental type thing, but is it more like dealing with heredity, genetics? That's why some people are more prone to get PTSD soldiers.
It's definitely environmental, but a large part of it is maybe due to some genetics. Some people may be more likely to get it than others.
So it's then genetics probably plays a part then.
Yes.
So our project addresses both neurological and psychiatric disorders, meaning PTSD in the psychiatric disorders. So essentially, even though it may be having to do with genetics and it may be having to do with environmental effects, we're essentially using the peptide probes for the same thing. We're trying to identify what is going on in the brain and how it affects the person as a whole.
I think we have time for one question from our instructors or TA, and then Erica can answer. Can you read that question?
Maybe. I would have been interested in understanding how the peptide probe actually is specific to, how do you make the probe specific to a specific disease?
Okay. So there have been studies in Singapore, in a Singapore consortium, making new models for these probes to have the specificity to identify and make sure that what we're trying to look for is exactly what we're getting. And so using these new models of the probes, we'd be able to identify a gene from a gene and a chromosome from a chromosome with no mistake for switching them up.
Thank you, guys.
Thank you.
All right. Next group.
Hi, good morning. I'm Holden Orius.
I'm Albert Gonzalez,
I'm Noah Schechter.
and we will explore about using fluorescent pulse neurotransmitters to map neural connections.
So we look to determine the synaptic level neural transmissions of serotonin in the brain, especially in the occipital lobe. And to do that, we will take live brain tissue cultures, and we will use a fluorescent false neurotransmitter, which is a fluorescent dye that mimics the structure of a neurotransmitter. So it can be imaged under under confocal or Sted microscopy, which was discussed earlier. We will also excite a neuron to watch how the pathway works in that sample of brain using optogenetics, which is a field of exciting neurons using light.
So after looking at our research papers and other sources that we found online, we decided to pose ourselves this specific research question, which is, what specific neural path does serotonin take in the occipital lobe of mice at the synaptic level? And the knowledge potentially gained from answering this question would contribute to brain mapping in mice. And since the brains of mice are basically scaled, a watered-down version of human brains, the knowledge gained from this could potentially contribute to the central goal of the BRAIN initiative.
And this is a long-term social context, but understanding the exact synaptic levels, synaptic pathways, through which serotonin travels in the occipital lobe of mice could eventually contribute to a larger understanding and mapping of the brain of both humans and mice, which at some point in the future, not only accomplishing one of the main goals of the BRAIN initiative, it can also lead to advancements in medicine and technology, which can save lives and money.
So to complement our work, we looked at some studies and we selected the most important information. Scientists are able to prepare live brain slices from adult mice for observation in a microscope. The fluorescent false neurotransmitters, the FFN, are currently being used to image and study synapses, and the transfer of dopamine has been studied using FFN in the past.
So after research, we decided to propose this for our project, which would be A, to introduce a fluorescent dye which would simulate the structure and function of serotonin, so it would be able to be passed along through synapses when neurons fire. Secondly, we would integrate the FFN into a living brain culture of adult mice, along with an optogenetic actuator. Now, you might be thinking, what the heck is an optogenetic actuator? And what it is, when you stimulate neurons using optogenetics, which is basically a field of science that uses light to get neurons activated, you need to introduce a specific gene that knows when the light's targeting it, so it'll activate the neuron. So we would optogenetically stimulate the neuron while it's under Sted microscopy, and using this, we would observe the pathways of neurons as the FFN travels across each synapse. The advantage of using Sted microscopy for this is that combined with double lasers plus a pinhole to block out excess light, we'd be able to get very specific, high-resolution 2D cross-sections of the brain sample all the way through, so we'd have a better map to analyze in the end. And in the end, we'd be able to map the pathways the false serotonin takes as different neurons are stimulated.
To reach the final objective, we first need to surpass some obstacles. Optogenetically stimulating a neuron under Sted Confocal Microscopy is a new technology, so the knowledge about it, it's not quite enough to solve some issues that could appear while observing the sample. The Sted laser and the confocal laser must stimulate unwanted neurons, what would not help in mapping the brain. And it's difficult to prepare slides for Sted because they need to be really, really thin, and the thin brain slides have a few neurons, and that could not be enough to completely map. Also, the Sted laser and the confocal laser could have enough power to kill some of the cells.
So in summary, the goal of our research would be to study the neural pathways of mice in the occipital lobe all the way down to the synaptic level, which hasn't been covered in too much detail before. And the project would use a fluorescent pulse neurotransmitter and Sted confocal microscopy to track the firing of neurons releasing this neurotransmitter, serotonin. After getting the results, we'd be able to possibly conduct future analysis in other parts of the brains of mice to further create a comprehensive map of the mouse brain. And the results from this, we'd be able to extend to human brains because they're very similar in structure and function.
So we all contributed to a variety of components of the PowerPoint, and we credit various scientific literature and other informational text.
Thank you. Any questions?
All right, questions? Yes.
Just like traffic here in LA, are there like possible bottlenecks in the neurons?
That's
Is that what you guys try to prevent, or?
We're not actually trying to prevent any certain disorder. What we're trying to do is map on the synaptic level how the serotonin is transferred between one neuron and the next, and how the neurons signal to each other, because that has never been done before for serotonin on the single neuron level. So we're not trying to necessarily treat a disorder that could come further down the road.
Is there a disorder that bottlenecks like the neurons? Is there like a bottleneck that causes a disorder that does that, or would that be in your field?
That's beyond the scope of our project in particular, so I don't know.
There's a question over here.
And over there.
Question over here? Yes.
Why serotonin? Where does it connect to in the brain?
Okay, so when you're mapping neurons at the synaptic level, there have been many studies done targeting, say, dopamine and other neurotransmitters as such. But in the occipital lobe, one of the neurotransmitters that's more in action than dopamine is serotonin, and so we decided to target that instead.
Instructors, TA, questions over here?
I want to read your paper.
Yes.
You mentioned that there's a live brain slice.
Okay.
What is
So-
that? I've never heard of that before.
So this is one of the things we actually managed to discover in the scientific papers we had. When there's special ways to prepare slices of mouse brain. It's not say completely like alive and functioning inside the organism, but it's able to be preserved in culture long enough for the neurons to still have their function.
Thank you guys very much. So since you guys
have been sitting here for about an hour. We're going to take just a very brief 5-minute break. So stretch, go to the bathroom, do what you need to do. But let's get back here in 5 minutes because we still got 5 more presentations. All right, so next group, are you guys ready to go? All right, so if everyone would please take their seats in a timely manner, we're going to go ahead and get started with our next presentation.
Hi everyone, we did our project on using microscopy for detection of signals in neurological diseases and understanding the human brain. So I'm Emily Dew.
I'm Naya.
I'm Dryly. So the brain is mostly a mystery to scientists, despite the improvements in the technology that is available in the current day, and the great number of people who work in neuroscience. As a result, we find ourselves ill-prepared to treat many neurological diseases, many of which have unknown causes and detrimental effects. The brain carries out functions through the signaling, so we wish to understand how different types of signaling may be inhibited or altered in diseases. Developing a full comprehensive picture of the human brain and its activities could potentially help all of those who suffer from such neurological diseases.
So we wish to see how current methods of nanoscale microscopy can address neurological diseases such as schizophrenia. So we thought this was a relevant topic because nearly 1 billion people in the world suffer from neurological diseases, and as the world has a population of 7 billion, that's approximately 1 in 7 people. So in a social context, accurate and in-depth information from imaging can help us develop better medicines and possibly lead to cures for these diseases. So as there's a large amount of information that's unknown about what these diseases are caused by and how to possibly address them, there's a lot of fear surrounding these diseases, and additionally there's sometimes social stigma around them, especially around diseases such as schizophrenia or psychosis. And a better understanding of the brain can lead to better methods of addressing these in the future. So in the past, researchers have studied the activity of various neurotransmitters such as glutamine and dopamine. They've also studied genetics because there's been research on whether there are certain diseases that are hereditary or if certain genes make you more likely to develop a certain disease. People have also studied neuronal structure. So what we see is that in some diseases there are types of neural network breakdown, or in general the structure of neurons themselves are distorted or damaged. Additionally, people have used mouse models to study and simulate these diseases, and they've also looked at pedigree trees, in which case there are some diseases that are hereditary, such as Huntington's, and expansion microscopy to study brain tissue, and electron microscopy to connect images of the brain.
Okay, so for our project proposal, we didn't want to hone in on a specific method. We wanted to address all the techniques since the brain cannot be understood from a single perspective. So we've used all the methods that we learned this week and come up with a couple of techniques for each of them. So for optical and fluorescence microscopy, we can map the brain using clarity methods to get past the obstacle of opacity from the lipids in brain tissue. And we can use light sheet microscopy with fluorescent dye to study like single layers of the brain and then compile an image for that. And then we can also use expansion microscopy to study proteins and networks, and this has already been done by researchers at MIT, and they've studied that in the zebrafish. And we can also use confocal microscopy for deviations between healthy and diseased neurons. And for electron microscopy, we can use scanning electron microscopy to image slices of brain tissue and compile them into like a comprehensive model of the brain, which MIT has also done with the zebrafish. And for scanning probe microscopy, there are also a lot of applications to study the brain. And so we can use biological atomic force microscopy to study misfolded membrane proteins in neurons, since these have been linked to disease. And we can also put cells on the end of the probe and study neuron-to-neuron interaction. And we can also study the nanomembrane vesicles that we learned about yesterday to look for diseased cells in the blood-brain barrier, cerebrospinal fluid, to look for diseased cells sending out bad RNA and infecting other cells. And so for our challenges, we have two main ones, like ethics and technology. So for ethics, the brain is like a center for our personal identity and consciousness. So this gives us an obstacle as to where the line is concerning safety and privacy. And also theoretical breakthroughs can give us biological consequences. So for example, if we have a memory retaining breakthrough, then that could mess with the balance between remembering and forgetting, and that can cause a neural overload. And also cost and accessibility. So brain enhancements can, if they are developed, then they can widen the gap of socioeconomic status. And for technology, we don't have the technology available for us to know if there are more cells than we presently know about, like neurons and neuroglial cells. And there's also the issue of lipid opacity, which has been addressed with clarity. And there's also the issue of time and expenses, since these microscopic techniques take a lot of money and time. And so for our discussion, we wish to study signaling and identify patterns in neurological diseases and try to target causes and find cures for these causes. And so these present various questions, such as what are the predicted possible irregularities that cause neurological diseases, and what correlations between symptoms and causes do we expect to see. And so these are contributions, and these are some references.
So when you said you were going to use optical microscopy in part to study the brain, you talked about using clarity methods. So like, what precisely does that entail?
Well, so the clarity methods have been developed. It's a technology where it allows us to see past like the lipids. And so, for example, like we saw like the picture in the PowerPoint, it allows us to see through to get the things we actually want to see. Like if we want to see individual neurons or networks, we can see past just the opaque tissue.
So are you thinking about trying to take brains, for example, maybe post-mortem, clarify them, and then look at, and image that entire brain or important sections of it in order to then compare the diseased brain to an undiseased brain?
Yeah, that's primarily our goal, just looking at the discrepancies between healthy brains and diseased brains. And since there's no ethical way to, say, image or model live brains, that's really what we have to work with, other than mice and zebrafish. Yeah.
One more question? Any other questions? No? All right. Well, thank you, guys. All right. Next group.
So I'm Matt Hashimoto.
I'm Cameron.
I'm Kyle.
And we decided to do our research on ALS observation and diagnosis.
So the main topic that we focused on is how can we better diagnose ALS at earlier stages? So ALS, or amyotrophic lateral sclerosis, is a very elusive and complex degenerative brain disorder. So scientists right now, they currently don't know much about what causes it. They have some theories about it's either protein, genome-based, but they're not really sure. So our approach is to not find a cure for ALS, but to find a way to diagnose ALS earlier. So the way we are going to do it is a protein released by a motor neuron when it dies is called a neurofilament medium polypeptide, or NEFM for short. So when it is released, we can examine it from people with ALS and people without ALS, and we can further examine it with using a fluorescent antibody tagging system and a light sheet microscopy. And if we are able to figure out how to continue with this research, we could save thousands of lives across the country.
Okay, and now on to precedents. So in current technology, scientists possess many methods to which they can image the brain and produce models and examine it to any extent. But it is a little bit limited as of now. That's why we have the Brain Initiative. And so some of these techniques are fMRI, CAT scans, and then they can also image dead samples. And let's see. So next, there are stem cells. So stem cell research is hot and new right now. It's been progressing over the last few years. And although they haven't advanced it enough yet to start using it to its full potential, but hopefully we'll see that in the future some near time. Still under stem cells are iPSCs, or induced pluripotent stem cells, which scientists can use to grow any type of cell in the culture and study it there. And lastly is protein analysis, which is the study of proteins in CSF and blood, which can be used to diagnose ALS, and I'll get into that more later. And then so on to our project proposal. So we propose a method for diagnosing ALS using a protein normally found in motor neurons, which he talked about earlier, which is called NEFM, or neurofilament medium polypeptide. And that protein plays an important role in intracellular transport, as well as just general structural integrity of the neuron. And so in ALS patients, what they've observed is that in their CSF and their blood plasma, there is an increased level of these proteins. And so the first step is to find willing volunteers with and without ALS for spinal taps. And to do that, we must take multiple samples. And then the second step is to tag the protein with an antibody. So the way this works is a two-antibody system. So one antibody binds to the protein first, and then a second fluorescent antibody binds to that first antibody. And so what that does is it makes it easy for the protein to be imaged with fluorescent microscopy. And we will use LSM, or light sheet microscopy, to accomplish this. And in the disease, we should see something like this on the slide here, is lots of fluorescent dots, and those are the proteins. And then normal patients, we should see little to no fluorescence. And then finally, the third step is to determine if the NEFM protein is the same in normal patients and in ALS patients. And this, we can either isolate the protein from the CSF and examine it, or we can take a biopsy of motor neurons.
And then about the social context. By researching a new way to diagnose ALS earlier, many lives can be saved. By researching a diagnosis, it brings us a step closer to understanding how ALS works and develops. With an earlier diagnosis, it may be easier to study the early development and stage of ALS. This may be able to provide the people living with ALS in the United States alone. Approximately 20,000 to 30,000 people have ALS, and 5,000 more are diagnosed every year. With the research that will be done, a cure or diagnosis indicator that will drastically help the society. Then about the challenge. One challenge may be finding willing volunteers for spinal taps. It's not a very fun procedure. To say the least, we may also have to try to obtain as a random sample as possible to preserve statistical accuracy. The test is not 100% for ALS, as other diseases may cause the death of neurons, and then, in turn, an increased concentration of NEFM.
All right, so future directions. So this diagnosis will take us one step closer to either finding a cure for ALS or finding a more complete diagnosis. So using our technique, we can use high-resolution imaging, such as light sheet microscopy or maybe even transmission electron microscopy, to further study a cure for ALS. So for example, if it turns out that it is a genetic modulation in the ALS cells that will cause it, we could, new biotechnologies such as maybe CRISPR-Cas9, which is a kind of protein that has been studied to alter genetically some stuff. If we can apply it onto this concept, we may be able to alter the genes of ALS symptoms and then maybe fix the structure. So in essence, we want to create a new type of early diagnosis for ALS. So we will use the protein NEFM, which is released during the motor neuron's death. So when motor neurons die, it's usually a symptom of ALS. So we hypothesize that elevated levels of this protein in the blood and cerebral spinal fluid can indicate that the sample may have ALS symptoms. So this, with the motor neurons dying so frequently, this can lead to major brain damage and muscle dystrophy. But muscle dystrophy is basically when the muscles shrivel up and become very weak. So we will use LSM and fluorescent antibody targeting systems to track them. So in conclusion, when we see lots of NEFM protein in the blood and cerebral spinal fluid, we can then conclude that it's most likely that there will be symptoms of ALS. So we will want to use this in the future to be able to possibly diagnose ALS earlier before major symptoms start to occur.
Thank you.
Thank you. Then here are contributions.
How is light sheet microscopy in this case particularly helpful for your study?
Okay, so light sheet microscopy, we can use it to study live cells, which is always helpful because the light is minimally invasive onto it. So if you, in a traditional confocal microscope, if you were to use laser, it would take like pixel by pixel, right? Go like this. But in the laser, it can go straight, so it reduces the amount of toxicity which will be exposed to the live sample. So in our case, if we take the sample from the spinal tap and compare the two from someone with ALS and someone without ALS, then we could examine the levels of the protein that we previously tagged with the antibody and see if it's consistent, the data is consistent, we could hypothesize that if there's elevated levels of the protein, then it could lead to more likelihood of ALS.
So I need to clarify, or I need you to clarify. So you said that this NEFM is secreted as neurons die, right?
Yeah.
So and it's present in cerebral spinal fluid and in blood.
Yes.
So it's not in the cells anymore, right?
No, because so the protein is helped in structural. So when the neuron dies, the protein is a polymer. So when it dies, it breaks apart into the individual monomers and can spread out throughout the system.
Okay, so if it's then not encapsulated inside the cell anymore, then with respect to the last question, why does it matter to use light sheet, right? Because you're not trying, the cells are dead.
Yeah.
And the protein's not in the cell anymore. You're just trying to detect the presence of the protein in the fluid, right?
Yes.
So in that case, does the light sheet add anything for you?
We could, light sheet microscopy is very useful in using 3D renderings as well. So you could take it layer by layer to examine the shape of the protein and see if there's anything different between the two.
Yeah, another simple question. Do you know what the different level of concentration, let's say a patient has ALS, right? Everyone everyone keeps saying no one wants a spinal tap. I agree,
Yeah.
right? So what's the relative level of this NEFM in like serum versus cerebral spinal fluid? Like, if it's in the serum or in the plasma, why is there just so little of it that you can't use that instead of CFS?
Well, let's see. So for cerebral spinal fluid, I guess is, I think it would be easier to maybe see the proteins in CSF. I'm not sure how to really explain it, but.
So like research is very like experimental right now, so we don't know much more about it because there's lots of complications and variables that will change the results.
It's most likely that the concentration of the protein is higher in the spinal fluid because this is where the source is. But it's as you mentioned, it's probably also goes into the further grades, maybe goes into the blood. And this is maybe a place where you can also try to sample it and have an early diagnosis, right? So you can imagine you doing both, right?
Do one more question, and this question back there.
Besides ALS, are there any other brain diseases that also release this protein, and how you differentiate between Hispanic comparison?
So like Cameron said, this kind of testing is just like one of the first steps in analyzing, because other brain disorders, when they degenerate neurons, they will most likely release the same protein. So then we can at least see that, oh, something's wrong here, so we need to do further testing.
That's my question. How do you know it's ALS versus the other brain disease?
Right now, we're not sure if it can. We know that we can use this protein to distinguish that there's something wrong. It's just ALS is one of the major applications that this could be found in, and that studies have shown.
Thank you, guys.
Okay.
All right, next group.
Ignore that. I'll just hide it behind the podium. Should I stand in the middle? Pretend it's a backward C. And other letters.
Hi, I'm Andrew Chin.
I'm Caden
Okay, and I'm Kevin Clausen, and this is our microbiome initiative relating to the gut microbiome.
Okay, so our project involved the microbiome within the digestive tract of humans. It's a very interesting part of the body because we don't know a lot about the microbiome within our digestive tracts, and there's thousands of different bacteria living within the digestive tract as well. So we know that there's different bacteria within it that can perhaps cure diseases that we get, or they can also cause the diseases that we get because they're harmful. So we chose to look at colitis ulcerosa, also commonly known as ulcerative colitis. It's a disease that is caused by unknown reasons currently, and it occurs in the digestive tract. Over 200,000 cases occur within the United States a year. So we proposed to use confocal microscopy to assist our observation of the bacterial structure and function to perhaps find the cause of ulcerative colitis, and we think that it will help greatly.
So the main question we're trying to answer is how can we apply microscopy? Is that right? Okay. Techniques to further understand how we can determine the specific microbes in the varied gut microbiome that causes colitis ulcerosa. So like Caden said, despite many tests to determine what the specific cause of colitis ulcerosa is, unfortunately, they haven't found much about what the cause is. So based on the confocal microscopy techniques that we will explain later, this is how we propose that we're going to address the issue of how to find the cause of this disease.
Okay, so first a little bit of background about ulcerative colitis. So it is an inflammatory bowel disease type. So it affects the inner lining of the colon and the rectum, so it's mostly the large intestine. And it is characterized by the ulcers in the lining, as well as diarrhea, bloody stool, and cramps.
Confocal microscopy, what mainly differs it from other kinds of microscopy is that there's a pinnacle at the end which allows out of focus light to be eliminated, therefore allowing the finest detailed structures of the specimen to be observed. And while, the cause has not been seen yet of this disease, current technology advances have allowed our abilities to understand these diseases in general to achieve advancement in learning about the microbiome in the digestive tract specifically.
So research has been done before, experiments have been done on trying to find the causes of different inflammatory bowel diseases. So they've done microbiota fecal transplants. One study was done on rats where they took the fecal matter of a healthy rat and they stuck it into an unhealthy rat, and the unhealthy rat actually recovered from its illness after receiving the fecal matter from the healthy rat. So they also use diet manipulation to try and make sure that whatever they want going into their subjects is controlled. They also use probiotics and antibiotics to try and counter different bacteria within the digestive system.
By one study done by the American Society for Microbiology, they tried to observe the different microbes within the mucosal surfaces within the gastrointestinal tract. And by doing this, they were able to use confocal laser microscopy. This is going to be microscopy to allow a determination of the cells within, because with confocal microscopy, they use the staining called the FISH staining, which stands for Fluorescence Institute hybridization. And with this, they insert fluorophore labels that allow the colors of different cells to be recognized. So therefore, groups of cells and different organelles within can be recognized based on the colors that were basically given to them and allow them to be stained and recognized. And with this, they can allow the localization of different bacteria and cells to be recognized within. So based on this study, they were able to see that within the colon there were not a lot of bacterial differences that differed within the count. So based on that, what we propose to do is that we are going to observe the structural and functional observations within colitis ulcerosa, and the way we propose to do this is by, in the digestive tract, they contain the T cells and then the possible bacteria that cause this disease. And so what we plan to do is label the T cells different colors and then stain the bacteria differently to see which ones were eliminated first. The ones that were eliminated, therefore, because this disease tries its best to eliminate the most harmful bacteria first, but overdoes it. The ones that it does first are the ones that have the potential to be the ones that influence this disease the most. And then therefore the ones that were not killed off, therefore have the potential to not be likely causes of the disease. And by doing this, the ones that were killed off, this allows us to narrow down our scope because there's like 1,000 species of bacteria within the gut to allow us to determine what possible bacteria may lead to the potential cause of colitis ulcerosa.
So there's a few challenges that are going to occur within this experiment if we were to do it. People have very different microbiomes because everyone's different. They grow up in different environments, eat different foods, so they have very different bacterias, and perhaps one bacteria could be harmful in one person, whereas in another person it could be helpful to them. So another problem is that there are thousands of bacteria species living within the microbiomes of people's guts, and to find that specific bacteria that may be causing a certain disease would be very difficult because it's going to take going through thousands of bacteria. Another problem is challenge is that experimentation on animals is a little bit harder because you would need to use humans instead, because humans and animals have a different ratio of different bacterias in their digestive systems. They can have the same bacterias, but they'll have different amounts.
So there's that.
The contribution.
So, other than finding a cause for ulcerative colitis, our project would bring up certain other questions. One of them would be because, like Caden said, there are thousands of bacteria within the gut, and hundreds of them we don't even know exist. So, in our study, we will find bacteria that we've never discovered before, and they may have the potential for causing other inflammatory bowel diseases such as Crohn's disease or other colitises. Also, a second question that would come up would be, since many bacteria within the gut are beneficial, such as lactobacillus, it breaks down sugars. We would be able to figure out how this bacteria would be beneficial to the gut, and also, since we know the structure of this bacteria, we would be able to find a cure for it. And building off of that, the gut microbiome is a complex biodiverse ecosystem with each individual organism having a specific contribution to the microbiome. So we would be able to figure out how this specific bacteria would contribute to the microbiome normally, and also why it causes this disease. And these are references, so now we're open for questions.
Alex? So basically, this disease, colitis, ulcerosis, compared to a lot of other diseases, many people have narrowed down the microbes enough in order to determine which ones may be the potential for causing this. But for this disease specifically, what interests us most was that this was more like unknown, and scientists have done many tests to determine the nature of the microbes within this disease, but unfortunately, there hasn't been much success into discovering that yet.
So you mentioned that a transplant of microbes can actually result in a curative effect. Do you see any implications of things like antibiotic treatments and possibly causing things like ulcerative colitis?
Can you repeat that question real fast?
Yeah, so you guys talked about an example where bacteria were transferred from a healthy mouse to a diseased mouse, and that actually led to a recovery of that mouse.
Yes.
So then, for example, when we take antibiotics and we wipe out everything indiscriminately, do you see a role in that potentially contributing to some of these diseases? And did you find anything discussing that in your literature?
Yeah, I actually did. So there was a study shown where what happened was they did use antibiotics on a subject, or I can't remember what animal they were using, but it actually died due to the antibiotics that they used actually wiping out everything in the microbiome that was in their digestive tract. And because they didn't have the healthy bacteria there existing to probably counter the harmful bacteria continued to multiply and just take over the digestive tract and kill it.
If there are over 1,000 different type of bacteria, how you propose or narrow down that this particular bacteria is the cause for ulcerative colitis?
So like the stain that we discussed, the fish stain, basically we'll have to if we ever try out this experiment, we'll have to see like how many like different species within our first like sample we see, and then we'll have to stain them each differently in order to determine how each of them are set apart, and then that's how we can determine from the T cells which ones are over time, which colors are noticeably gone because of those bacteria being eliminated first.
Do you remember anything from your tour on the first day up on the 6th floor in the MSSR lab with the robots? Right, the high throughput screening facility, right? So this is an area where addressing thousands, tens of thousands, hundreds of thousands of samples, right, where we're integrating optical detection, fluorescence detection with high throughput methods really allows you to address some of these bottlenecks, right? It's one of the reasons that we brought you up there, even though it's not a microscopy facility per se, it really overcomes some of these limits, right? Because the number of samples that you need to do these types of studies in certain cases is very high, right? And becomes cost prohibitive, right? So increasing throughput is a potential option to address what he's talking about, which I don't know if you guys remember anything from that tour, but maybe you could comment on it if you do.
Oh, there was another comment. So another way that we might be able to narrow down the scope is ulcerative colitis is an autoimmune disorder, so it does start to attack itself. And this is because the bacteria that potentially causes it, the immune system will attack it, but it doesn't stop attacking it. So in our studies, when we add the T cells to the bacteria, the T cells will just continuously attack the bacteria, while other bacteria, it'll just stop.
Have fecal transplants been used successfully in humans? No?
I didn't find any studies about this because I don't think anyone wants that to happen.
Well,
I thought I heard that it has been done.
it has been done.
I didn't find anything on that, so.
Cure's a cure. Well, there's a difference between what's been done and what's been published in the literature. There are certain clinical trials and methods that are in process in certain areas that haven't yet been published per se. So I think most of the students are leveraging either the web to look at the scientific literature, but that stuff is ongoing and has some pretty strong positive implications. It does speak though to the realities of people getting over what it actually entails.
Well, and eventually, if you could really identify things, you can always improve the treatment and provide just the bacteria, for example, right? But you have to learn that first. You start with the materials you have to work with, which right now, since you don't have these techniques fully in place, you can't isolate exactly which bacteria were the curative ones. So you work with some kind of bulk, person-to-person transplant using this material. Later, if you really refined and understood it, you can get rid of all of those details and just provide the curative aspect of what it was. You don't need to do that blunt force version of it.
That was just more of a comment. What was the disease in Africa where some people came here and that is exactly what they did, right? It was a fecal transplant records like last year. Yeah, that was the, no. They were really sick in Africa, most parts of Africa, and they stopped traveling. Do you know what I'm talking about? I just don't remember.
Oh, so I was.
There are, as some people have been discussing, there are really strong links between the gut immune system and the overall immune system. You think about your gut processes all sorts of foreign material that the rest of your body would freak out in exposure to it. And so how your immune system in the gut translates to your overall immune system is not fully well understood, but it's known there's very strong linkages. So it could be that you're basically activating some immune process in the gut that then actually then goes out and activates other immune processes or gives you antibodies that are developed in the gut that then can proliferate to the rest of your body and give you a cure for a virus, for example. So that's why it's very, very interesting to study gut immunology versus total immunology, as one of our first groups mentioned.
I got one question. This isn't really specific to this, but there's a lot of overlapping and all that stuff. I was wondering, is there like a cloud for this that people share, or is it so guarded that, like you mentioned about it's not being published or what and all that stuff, is it so guarded that they don't put it up in the clouds for other people to, you know what I'm saying?
Yeah, I know what you're saying. I mean, it raises a broader ongoing discussion in the scientific community about open access to the data that you all fund through your taxes, right? And that large publishing companies make a lot of money on keeping it behind paywalls, right? So the National Institute of Health, the NSF, major funding agencies are requesting and requiring that we make data available through open access repositories. The UC system actually requests that all of our pre-print publications are uploaded to a server that's available to anyone to use, but it's really something that's only been in the discussion in the last, meaningfully the last 5 to 10 so much scientific literature out there, and with scientific literacy of the general populace and interest increasing, and the sort of methods we have for sharing data online, there's a lot more demand for people to really have access to this stuff, but it's just not the traditional approach. It's usually behind, you can only get in if you're on campus of a university or if you have a subscription to a journal. That's slowly changing. There's a lot more open access publications. But the model is one that's under a pretty intense discussion in science in general about how to make the data available.
Yeah, because of the acceleration of AI and all that stuff, that could greatly help and all that stuff. There was like a cloud and all that stuff.
A lot of people are being much more open to sharing their data more broadly to leverage these sorts of, whether you talk about cloud-based crowdsourcing, scientific crowdsourcing, right? There's other approaches to things, but it's really an emerging area of people figuring out how to strike the balance between, making sure that the science is well vetted and peer-reviewed, but making it accessible not only to the general community, but also to other scientists in a way that doesn't jeopardize the individual research integrity, right? So it's a very vigorous dialogue right now, but it's something that's still not where you might think or hope it will be.
Another question for presenters? Yes, this is kind of on that vein. You keep talking about how spinal taps are so advanced and whatever else. Is there something sort of like, when you get your license, you're a body part donor or an organ donor, where when you're having one of these tests that's so vile that you would sign something so that the rest of the materials in that test could be utilized by other scientific communities, or is there anything like that?
There actually is a building movement to have people, and many people can donate their bodies for science. Oftentimes when
I'm
people
talking
do
about like
studies.
an active person who is having a spinal tap. Part of that fluid could be used for some others.
Yeah, so there's one where you're talking about like organ donation and and actually donating the whole body, right? And on the other side, yes, when you do procedures, you can sign off to allow scientific researchers to have access to those materials as well. So yeah, collecting samples from hospital settings and from medical practice is actually very it's like one of the things that powers a lot of research here at UCLA is we have a medical school. And so we can go get biopsy samples when they take out some tissue from a brain. Like they mentioned looking at motor neurons and doing biopsies there, right? When you do, for example, a resection during cancer, you will get some healthy tissue as well. And so dividing up those samples to researchers. Now the patients have to make sure they acknowledge and authorize that, right? And then of course their identities have to be protected, and there's all sorts of protocols on top of that. But that's definitely a well-established practice, and also something that's increasingly being spread to different types of tissues. And there's also tissue banks where people can send wholesale tissues, because some problems can only be addressed when you collect 10,000 tissue samples from diverse groups of patients with and without the disease.
So is it standard procedure that if you're having one of these tests that you're given a proposal to sign off on it?
It's
or is
growing.
It not,
Depends where you are. Yeah, and whether you're a research hospital or a regular hospital makes a huge difference oftentimes too.
All right. Well, thank you guys very much.
All right, group 9.
Hi, I'm Braden, this is Hank, and this is Sarosh, and we're doing the development and application of high-resolution imaging with regards to microbiomes with SEM and TEM microscopes.
So when learning about microbiome and the organisms that live within it, one of the greatest challenges is finding microscopes that are capable of viewing the extremely small organisms that live within at a high resolution. Two of the microscopes that are capable of doing so are the tunneling electron microscope and the scanning electron microscope. These microscopes both use a beam of electrons to scan the subject what you're looking at under the microscope. However, the scanning electron microscope only looks at a topographical view of the subject, and the tunneling electron microscope uses electrons to pass through and create a more in-depth image.
Now, what we were trying to find out is what is the better microscope for researching in the microbiomes, the SEM or the TEM, and how we could possibly improve these methods.
By knowing which microscope is more capable, we could come up with new ways to improve their respective resolutions in order to better understand the chemical and physical makeup of whatever you're looking at.
Now, microbiomes are in a lot of things, from the human body to global precipitation and agriculture, as shown in the slides. And it is extremely important in medicine, because if we figure out how the microbiomes work in the human body, we could pre-diagnose certain diseases and figure out how they work and possibly cure them.
Okay, so now currently scientists, to study the microbiomes, they're using two types of electron microscopes so that they can see the smallest objects. They're using the SEM and the TEM. So the SEM is the scanning electron microscope. Now, some of the advantages of this microscope is that it's easy to use and takes a topographical image, which means it only shows like the top layer, so it's good for studying like the surface and the shape of that, and it requires minimal sample preparation. But on the flip side, it only images the surface, which doesn't show us the inside of the organism we're trying to study. So if we want a more in-depth view, we should be using a TEM microscope. And it's also very sensitive, so we have to take special precautions to make sure there's no extra vibrations in the environment. And you also have to get a table that can get rid of those vibrations. It's a thing. It's an IQ damn big table.
Yes. Now, the TEM, or the tunneling electron microscope, is easy to use just like the SEM. And it is very easy to convert the images to a 3D model. And the magnification of the TEM is 5 million times what the naked eye can see. We can take both large and small sections of the subject, so if we want to see a group of cells or a single cell. And we can also view inside the cell. But on the other end, the technology is very expensive. And it's very tedious and time-consuming. It can take anywhere from a couple hours to 20 hours to produce a single image. And it requires extremely specialized training to work these microscopes. And the samples have to be extremely thin for the electrons to pass through. And the samples have to be able to tolerate a vacuum chamber so they don't get damaged. And the images also render in black and white, so it's really hard to see color.
So now our proposal has to do with increasing the resolution of the images produced by the TEM microscope, which we decided was better for this purpose because it shows the inside of the cell. So every object vibrates and the molecules are always moving around. So when the microscope wants to take a scan or an image, it's obviously hard to do when all the molecules are moving around. So what we want to do is try to slow down the molecules without damaging what's inside the cell. So right now there's a method called Cryo Scm, where basically they freeze the cell, but the problem with that is we can damage the cell and the bacteria inside of it, which makes it hard to study. So what we did was we looked at a study conducted by Northwestern University that uses a beam to slow down particle motion. Now this beam is a broadband laser light that has multiple frequencies. What the researchers did was take out all the frequencies that would increase the molecular motion and only left the ones that would decrease it. So therefore, they're able to cool it without damaging the molecules inside. So now, they've already done this before, and it's been used and proven that it works. So as you can see on the slide, there's a case study. It was used to cool aluminum monohydride molecules from room temperature to 4 Kelvin, which is almost a 290 Kelvin difference in less than a second.
Now, the challenges that we're going to face are that introducing a cooling laser is very hard in a TEM because it could introduce new particles like photons, and it could disrupt the images because you need reflection to get the image. And the cooling could also possibly damage live cells prematurely. Plus the introducing the cooling laser into the TEM is very expensive because the TEM alone is roughly $500,000 to $1 million.
With new methods in science comes change, and if these techniques become relevant, nanomicroscopy would change. With a new understanding of the microbiome, the only plausible decision would be to take the new information and use it to its full ability in order to understand the way microbiomes work and how everything inside works.
Now contributions, we all worked on it. And then references. Okay.
Laser cooling approach that Northwestern's taken is pretty slick. It's only been applied to very narrow case studies, right? And I'm wondering do you think that it would be applicable to a biological sample, right? Like in this, based on, they were doing a very simple inorganic system that demonstrated basically a physics experiment, right? And that's where many developments in measurement microscopy start from. The question is, can you apply it to a biological structure, right?
I think that if applied in the right way, as in Cryo Scm, you have to do it in a very specific way in order not to damage the cells. I think that if it were to be done in a way that didn't damage the organic molecules, it could work, but that would also be difficult and require some research in order to pull off. So it just depends on how much time you have.
So you talk about cutting edge technologies and stuff that have been developed recently. But so for your project, are you like combining those technologies together, or like are you making improvements on second technologies, or how are you using those to like advance your project?
Basically what we're trying to do is improve the resolution of the images. So right now, both the SCM and TEM obviously do great work. But the biggest issue is that with the molecules always zipping around and vibrating in solids, it's hard to get a clearer picture. It works the same way on the micro scale as in like on the macro scale. If you're trying to take a picture of a ball flying through the air, it's hard to do. But if we're able to slow down that motion, we are able to get a deeper image and we're able to see more details, which will help our studies.
We know that a lot of scientific tools and scientific methods and discoveries are proprietary. I mean, we just talked about that. But are any of these, any of the people working in these different places, are they working together to, I mean, that's obviously a new method that they were using. Is there a way to work together with the tools and with the scientists? I'm assuming there are different types of scientists working on the different tools to make them, to sort of elevate it at the same time, rather than it seems like they tried this, it sort of worked, then they're going to try something else, it's going to sort of work, so that it comes together at the same time moving forward? Does that make any sense?
Okay, I got it. So yes, it makes perfect sense. Like the issue really isn't like proprietary interests, but it's more about the fact that this technology can be incorporated into nanoscience in order to help us produce clearer images. So like it's not a matter of which one is better, but a matter of using that kind of technology to further a different branch of science.
She also asked if like scientists could work together to further it.
I wouldn't know if scientists could work together on that. Like that's a proprietary issue that's between them, honestly.
I think the bigger issue, we do a lot of instrument development here, is that historically you tend to get technology in searches of problems, right? You get people making things that are really cool and they can do something, but it doesn't have a sort of scalable, broadly applicable purpose. And I think what's shifting over the last, 10 to 15 years or so is really bringing the people developing technology together with the people doing specifically, interesting research areas to develop technologies that are specifically designed to address a need, rather than just making some cool new gadget that produces a paper and a news story, but then doesn't ever have any real world impact. Right? So part of what this course was about that the students read about was the need for new tools for the microbiome, the need for new tools for the brain initiative, right? Like purpose-driven instrument development, not just developing a technology for the sake of showing you can do something fancy, but to do it in a way that actually addresses a real-world identified need from scientists, from physics, from chemistry, from the medical field, and everywhere in between. And that's where nanoscience, through its sort of interdisciplinarity, really lowers the barrier to that happening. Because in this building, we've got people that have real problems, and we've got technology development, and they're all working and collaborating together. So I think that what you're pointing out is a lot of is true historically. It's the mindset is shifting a bit as you start to sort of break down the silos of traditional departments and you get people talking to one another. The proprietary side is always a tricky one, just because we do necessarily work with companies at some level. I mean, if you wanted to add this technology, for example, to RTEM, that's a $10 million microscope made by a company. You know, we would have to collaborate with them, and there are balancing acts that need to be struck when you do that. But it's certainly not anything that we don't attack if it's necessary.
And is science still going towards development if it makes money, or are we now finding that these advancements that these amazing students are looking into, that it's enough that it will help society and not just make a load of money for some company? Are we getting there yet?
It's a tricky one. I think that actually it's been historically the other way around. You saw more basic research happening in the last 50 years than you tend to see now. The shift tends to be more now towards actually applications-based or translational science, where you have to be addressing a particular problem, like the willingness of the funding agencies to support basic discovery-based science, especially in the United States, is not as vigorous as it has been in the past. A lot of companies don't invest in basic industry, research and development. So it definitely goes through ebbs and flows. Whoever's in Congress and is deciding the budget affects the Department of Defense, Energy, NSF, NIH, CDC, all the funding agencies. So the mindset of the federal government also affects these things. I think, I don't know, Laurent, what you would say, but it seems to me, at least in the last handful of years, that a focus on application has been on the uptick, more so than basic science. That's not necessarily only in the perspective of sort of working with or enabling companies to make money. It's that people are asking, what's the impact of this science that we've been investing in, especially when it's hard to see.
Yes?
About stopping motion and all that stuff. I'm a film photography major. A lot of ways of stopping motion is to move the lens hand with the action. So instead of like freezing the molecule, or you could develop an instrument that can move with the subject matter. I know that's going to be impossible. Well, right now, theoretically, it's impossible because you don't know how you're going to predict movements of molecules or whatever. But you could develop an instrument that moves with it, then you're going to get more sharpness with it because you're moving relative to the speed of that molecule. So that's one approach for you guys to think about.
Yeah, I mean, that's definitely one way to approach it, but I think that molecules move in such an erratic pattern that would be quite hard to develop tracking software that could predict its movement. So.
From stopping its motion, you could also go to controlling its motion.
Yeah.
And maybe instead of freezing the water, have it move up and down at a predictable frequency that then you can compensate for with your equipment. So this idea of using a laser to adjust molecular motion has applications besides just stopping it. So I think it's very interesting and innovative, cutting edge technique.
All right, last but not least, Group 10.
Okay, our focus today is fluorescence microscopy and Parkinson's disease. And I'm Cami.
I'm Mackenzie.
And I'm Laisha.
Okay, so ever since the first published academic paper on Parkinson's disease in 1817 by Dr. James Parkinson, scientists have been furiously researching how this neurodegenerative disease takes over the brain and the motor functions of a human affected by it. As scientists continue to struggle with finding the cause of this disease, they began to study the changes the body of an infected person goes through and discovered that the brain severely decreases its release of dopamine after an infected patient reaches around 60 to 65 years old. Despite knowing the symptoms of the disease, the scientists have not been able to figure out why the sudden decrease of dopamine in the brain occurs. Recently, scientists began studying the brain using microscopes in order to search for ways to cure this disease or for a stronger and more effective medication to lessen the symptoms.
Okay, so our focus question is, how can fluorescence microscopy help advance current research in Parkinson's disease? And before we go into the answer to that question, first I think it's important to talk about fluorescence microscopy. So it's a general term for a really complex concept, but it's involved in a number of techniques that benefit the scientific community. But the principle of it is applying colored dye to subunits in order to help distinguish their locus or their function, which is a really important tool in comparison. So the process works by adding a source of light in order to excite electrons to a higher energy state, and the level of which dictates the color that they show. And fluorescence, by this principle, is defined as the emission of light by a light-induced singlet, and that singlet would be electrons in this situation. And so Parkinson's disease is a neurodegenerative brain disorder, and the problem is that we don't know the root cause of it, but what we do know is that damage in dopamine-producing cells leads to the levels of dopamine dropping, which ultimately results in the symptoms of this disease. And there is some evidence today that shows that it is a genetic disorder, although it is not solid.
Okay, so the social impacts of Parkinson's disease. In the United States today, there are approximately 1 million people affected with Parkinson's disease. It involves the malfunction and death of vital nerve cells in the brain, and therefore symptoms worsen over time as the patient gets older. Some of the most common symptoms includes tremor of the hands, the arms, the legs, the face, and the jaw, slowness of motion, stiffness of limbs, and impaired balance and coordination. When nerve damage gets too severe, surgery may be necessary. However, medication is typically used to help lessen symptoms. Although scientists are unsure of what causes Parkinson's, some speculate that it is related to clump proteins in the brain. And these proteins, there are two of them that we kind of talk about. It's alpha-synuclein and proteins from the gene LRRK2. If we can isolate these genes and image them over time, we may be able to create a drug that helps suppress the translation of the genes that encodes for these proteins and prolong the lives of the people with Parkinson's disease.
Okay, so some background and precedent on the issue, or on fluorescence microscopy. It was first defined by Sir George G. Stokes, and he coined the term in the year 1852. But further research throughout the 19th century confirmed his belief that organisms will fluoresce when put under ultraviolet light. And another important principle about fluorescence microscopy, and what may even perhaps be considered a limitation, is Stokes' law, which states that as the electrons come down from their excited state into a more relaxed state, energy is lost, and therefore it must be compensated by a shift in the light emission spectrum, which results in longer wavelengths. So we can compensate for this by using fluorescence filler combinations, which will kind of help us separate between excitation, which emits light. So our project proposal to sort of delve into the issue is working with the LRRK2, which is a gene that encodes for a protein found frequently throughout the brain. And it has been seen in the past to be linked to Parkinson's. So in past cases, they found that mutations of this gene will result in the damage in dopamine-producing cells. So our idea is to use a technique which is called fluorescent in situ hybridization, which stems from the concept of fluorescence microscopy. And it combines principles from both light microscopy and molecular biology by first targeting the DNA sequence of interest, which is the LRRK2 in our situation, and to then track which parts of the brain is found, use the fluorescence to stain these parts, and then we can see their location and make some important comparisons, which will hopefully guide our investigation into the cause.
So the BRAIN Initiative started in 2013. It was brought up by our former President Barack Obama, and what basically scientists wanted to do was look more into diseases that were from the brain, which is like Alzheimer's, Parkinson, and other diseases. But some of the obstacles scientists have faced are that the brain is way too complex for them to look at, and it is too big for them to look at it completely, like altogether. And it is also the technology they use is not far advanced for them to look at the brain, and because they can't look at a brain of a person who is alive, it is more difficult to them to find what they need. So in order to advance research, what we're gonna do is we will color dye highlight particles. We'll highlight particles to see their location, and this we will use fluorescence microscopy.
Okay, so some important discussion questions surrounding the topic. One is, in the case that Parkinson's disease is found in the subject's genetics, what are some ethical concerns that can arise developing solutions for prevention or cure of this?
A second question that we have is, how would you be able to determine which protein is causing the decrease in dopamine, if it is in fact a protein? Because scientists aren't really sure right now what protein it is, or if it actually is the protein that's causing this. And third question is, who funds projects like this, and what are their motivations for doing so? And also, how much money should go into this research? Contributions. Yeah.
Questions? Yes.
So how does Parkinson's develop throughout time?
Okay, so basically what starts off when people, it's like different for everyone, but if someone is infected with Parkinson's disease, it normally becomes prevalent around the age 60 to 65, and it starts normally off with like the shaking of the hands, and then it keeps progressing to like other shaking, like stiffness of the body, just like lack of coordination, and eventually as the vital nerves in the brain keep dying because they don't get the dopamine, some people require surgery. Like, there are medications that are used to suppress these symptoms. However, sometimes they don't work. It's different for everyone. And so if they do need surgery, it's called deep brain stimulation surgery. And basically what it does is it blocks electrical signals from target areas of the brain. So it kind of like focuses, it kind of like, it just basically, it like doesn't harm healthy brain tissue by like destroying the damaged one. So they have to do that. Yeah, so basically as they get older and older, they start to lose more and more function. It could also lead to neuropsychiatric disturbances, which could be disorders of speech, cognition, mood, behavior, and thought. So yeah, it kind of progresses over time to it keeps getting worse and worse as more vital nerve cells in the brain die.
I know we don't know the root cause of it, but is there any information on what damages the dopamine-producing cells or what could possibly cause damage to them?
Sure. So I think what I think the problem that they find is that the cells are no longer producing dopamine. I think that
There's a photo. So if you look at the top, that's the normal neuron and neurotransmitter, like the amount of dopamine, and in the one below it, that's after, like when they're affected by Parkinson's disease, that is the change, I guess. But yeah.
Yeah, I think they don't know what's causing damage. They just know that they're not producing anymore. So it's probably somewhere along the tract where cells are emitting proteins. If that makes sense.
You mentioned at that point it's like the clumping of cells, or what, you mentioned something about
Yeah.
clumping.
It's the clumping of certain proteins. That's what scientists today are currently looking into to see if that's the root cause of this lack or this decrease in dopamine production. Like, they're still trying to figure it out, but there are various proteins or clumps of proteins that show up in people who have Parkinson's disease, and so they're trying to see, is this the root cause of it?
Where's it clumping at? The nerve ending?
I think it's just in the brain. I'm not 100% sure where it's clumping.
Yes. Speaking to your challenges questions, one of them was, what are the ethical considerations in developing a cure? And I was curious as to what those might be now, because I remember probably before you were born, when Michael J. Fox was diagnosed with Parkinson's, everyone was talking about the ethics of stem cell research, which is now part of the discussion here today. It's become part of the process. And so I'm wondering what else is currently at stake.
Okay, yeah. So I think there's the problem that other groups discussed, that testing on humans is really risky and dangerous. But then a whole separate entity is kind of altering DNA, because if we find that Parkinson's is caused by sequences in DNA, then that does give us the power to go back in perhaps an embryo and alter the DNA sequence in order to prevent Parkinson's from occurring in the lifetime of the child. But some people have religious concerns with this, thinking that it is unethical to alter DNA, and even though we may have the ability to do it, it could malfunction and it might be very risky or potential to harm the zygote's life.
So you said that the proteins basically come together, and then for whatever reason, the dopamine becomes non-existent or it's no longer there. If you could increase the amount of dopamine just by a pill, or there's pills now, do the clumps of protein not allow the body to store the dopamine? So if you could just increase dopamine, would that do anything good, or would the body still just reject the increased dopamine?
Yeah. Yeah, it really honestly depends on each person, because I know I read that the medication does really help some patients who have Parkinson's, because it does increase their dopamine levels, maybe not back to how it normally is, but get it back close to that. However, it does sometimes not work. I'm not 100% sure if it's the proteins or not, because they're not 100% sure that's the reason why dopamine levels dropped. However, I do know that does help with some, and with others it doesn't. And it really just depends on the patient, how they respond to the medication.
So with identifying the locations and regions of highest interest, do you think that would be able to help you identify later what is wrong with those particular cells by zooming into them? In other words, finding the right cells that are the cause of the disease versus all the other cells that aren't really participating in it?
Yeah, I think that could definitely help. especially because we know that there are a lot of different types of cells in the brain, not just like in function, but in purpose. Those are kind of the same thing, not just in location, but in function. So I think once we track the kind of cells that are not emitting the protein or that are maybe not doing something they're supposed to do, it'll help us like track Parkinson's development, but also help us kind of with the brain mapping as a whole.
I just got a question. Now Parkinson, it's like the neuron die, or they're like misfiring, or they're not communicating with each other. Is it like bad communication, or is it death of a neuron?
It's like a neuron's failure to do its job or like produce a certain protein that it's supposed to be producing.
Okay, because there was this other group that was talking about the flow of neurons or whatever. Do you guys work with them and develop new ways of healthier ones communicate with each other or something like that? I don't know. That's just my
Yes, I think that's also really difficult to do because then you'd actually have to go into the brain, and that is also like one of the things when you do have surgery, you're trying to like redirect away from the damaged nerve cells to the like healthy ones, and so I think that's part of it. But it's really kind of hard to tell without like going in and like monitoring the brain for for a certain amount of time.
All right, well, thank you, everyone. Good job to all of our presenters today. You guys did an excellent job of presenting the work that you did. Yeah, give them a hand. And you fielded a lot of very different types of questions, which is not an easy thing to do. Speaking as a grad student that gave a lot of presentations in group meetings with very intimidating professors, I can attest very personally to how difficult that is. And you guys all did extremely well. So once again, just great job to all of you. I wanted to take a minute to let our Executive Director of CNSI, Sonia Luna, say a few words. She very kindly agreed to come and just say her hellos and her thank yous and her congratulations. So please, let's welcome Sonia Luna.
I first want to apologize for not being able to spend more time with you this week. We've had a very busy week here at the CNSI. Lots of really terrific projects are starting to take root. And over time, I hope we'll be able to share more of those developments with you and the rest of our community. But I did want to just say a few closing words about how important it is for us to host you as our guests to the CNSI, the parents, the students, who have participated in this program. We have a firm commitment on education and outreach at all ages and at all levels. And as you can see from the last week of content that you have been through, there's just a wide variety of things that can evolve out of the work that we're doing here. As I was sitting listening to the last group presenting on Parkinson's disease, two thoughts came to mind. First of all, I would think that some of the work that you've done here in the last week was very personal. I understand there was a presentation about Alzheimer's disease. That to me is something very personal. My father had Alzheimer's disease. And so every day that I interact with people who can either bring new thoughts to the forefront that help address these diseases. If I'm in a room with a faculty member or a highly regarded researcher, I always sit up and listen to what they're telling me with regards to developments towards treating and curing such diseases as Parkinson's disease, Alzheimer's disease, we have a faculty member who's working on a treatment for pancreatic cancer. So as you move through the rest of your science career, I encourage you to think about how it affects you personally, how that might give you a trajectory on how you spend a meaningful part of your life and the work that you'll undertake over the next few years of your studies. And second, I kind of had to reflect back, I hate to say it, like 40 years. So when I was your age, the students in the audience in high school, and the type of opportunity that you've had here to stand in front of people and defend what you have learned over the course of the last week, that's an invaluable experience. You may not know it until way down the line, but when I was your age, I had to do the same thing at a science fair, and that early exposure to the discourse and exchange about scientific ideas is a really important skill. And Rita touched upon that as she was giving her remarks. It's not an easy thing to do. You're on the spot. You may not know the answer off the top of your head. I always think that the answer I don't know and I'll go find out is a really good answer to have if you don't know the answer. But as, again, you progress through the rest of your studies and hopefully a career in science, you reflect on this day and the things that you not only learned from a technical standpoint, but from the experience and the exchange that you've had with your colleagues here in the room. So again, we thank you so much for joining us. We hope that this was a beneficial experience to you, and I wish all of you the best. Thank you.
Thank you, Sonia. So it is my understanding that Mark, who is our silent but very helpful AV director in the back, has prepared a wonderful slideshow of all the times that he's been hovering around you, taking photos of you somewhat surreptitiously. So as we take just a little bit of time to sort of relax and regroup after having these presentations, Mark's going to play the slideshow. We'll have some music, and then hopefully soon we'll have some food and refreshments out for you in the lobby for you guys to celebrate the fact that you have successfully completed this course. So well done, everyone. Oh yeah, so I guess as this is going, what would be great for us to do is for you guys all to come up, and we would love to take a photo of all of you guys together to commemorate all of you being here and your hard work. So students, if you would please come up to the front and assemble yourselves.